All right, welcome. Wow, welcome back. Good morning. My name is Lengyi Cheng. I'm the Vice President, Head of Cell and Gene Therapy at Cencora. Welcome to the first Scientific Slam here in Rome. So we have a very exciting panel today. It's all about solid tumors, which is very timely given the first TIL therapy approval in the US earlier this year. So without further ado, I'll introduce our first speaker, Dr. Madan Jagasia, CEO, Obsidian Therapeutics.
Thank you, Lengyi, for the introduction, and thank you, everyone. So I'm Madan Jagasia, CEO of Obsidian Therapeutics, and it's my pleasure to share with you our journey in Obsidian as we take on the world of solid tumors. So as you look at cell therapy and solid tumors, the first challenge the solid tumor poses is that it has a diverse array of neoantigens. And these neoantigens are typically not shared across patients. So if you try and identify one antigen, which is sort of lineage specific, you're probably going to run into problems of escape because other neoantigens will escape. The advantage of tumor-infiltrating lymphocyte is that these are natural immune cells that the body has deployed in the tumor microenvironment. So these cells have already fundamentally solved two problems. They have already recognized a neoantigen, and they've figured out a way to get to the tumor microenvironment. But once they get to the tumor microenvironment, the cancer cells render them energetic through various checkpoint pathways. But if you were to take a piece of the tumor out, coax these tumor-infiltrating lymphocytes, and expand them and amplify them, you should be able to see the efficacy. And this is the current world of non-engineered TIL cell therapy. As Lengyi mentioned, we had an approval earlier in the US from Iovance. Their product is Amtagvi. The challenge is that suboptimal persistence and suboptimal expansion of non-engineered cells will create a barrier on the efficacy. So at Obsidian, we are leveraging our platform called CytoDrive to pharmacologically regulate membrane-bound IL-15 to improve antigen-independent expansion and persistence, which should translate into increased efficacy. So here's how we do it. So for our lead asset, OBX-115, the gene of interest is membrane-bound IL-15. It's brought under the control of what we call a DRD, or drug response domain. The drug response domain encodes for carbonic anhydrase. So this transgene is transfected into the TIL cell. The ligand for this DRD is an FDA-approved small molecule called acetazolamide. For those of us who have gone to high altitudes, climbed mountains, we've taken this pill for altitude sickness. This is also approved as a mild diuretic. In the absence of the ligand, this fusion protein of the DRD and membrane-bound IL-15 is degraded by the proteasome, and the basal state is what we call a drug off. But when you expose the cells to acetazolamide, either in manufacturing or give acetazolamide to the patient, you will see expression of IL-15. So here is a potential mechanism of action, walking you from left to right. The patient gets the lymphodepletion, gets the drug product, OBX-115, gets the ligand. The membrane-bound IL-15 is expressed. The way we've engineered the molecule is that the IL-15 can signal through its own cell surface receptor, or it can present the signal to a neighboring immune cell. Let's take the cis activation, where it's presented itself to its own receptor. You get normal physiologic effects of IL-15 signaling, which are antigen-independent expansion and persistence of the TIL cells. The neoantigens are recognized. You get direct killing of tumor cells. An important aspect of IL-15 signaling is that you get memory TIL formation, which drives the persistence of the product. So 2 slides on some preclinical data. This is basically showing that when we engineer the membrane-bound IL-15 and it engages its own receptor, you see augmentation of both STAT5 and S6. You see phosphorylation of these transcription factors, and this is modulated by acetazolamide and almost equivalent as if to giving recombinant human IL-15 to the cells. Now, interestingly, this membrane-bound IL-15 can also present a signal to a neighboring immune cell. In this model system, we have natural killer cells, and it can show that the NK cells, which we know respond to IL-15, you can also see an augmentation of STAT5 and S6. A word about manufacturing. Obviously, the starting material is tumor. We have optimized the first phase of manufacturing, that is called pre-rep or pre-rapid expansion protocol. We have an ancillary drug substance which is not present in the non-engineered platform, and this allows us to achieve two things. One is it selectively captures the T cells that have seen the neoantigen in the tumor microenvironment. The same drug substance allows us to start manufacturing from a core needle biopsy of the tumor, and we do not necessarily need a surgical excision of the tumor. So this allows easy access to tumor material. The activation and transduction is fairly standard. During the rep phase, we have a variety of differentiators. Our transgene is already in for the cells coming into the rep. We do not need IL-2 during the rep phase because we add the ligand acetazolamide, and this drives the IL-15 expansion. During this phase, the cells need a co-stimulatory signal. In the non-engineered platform, this is done by third-party mononuclear cells, which have been irradiated. We've taken it a step further. We've got a cell line that is expressing both IL-21 and 4-1BB, which is then irradiated and added to the media. So all of these differentiators gives a product its unique phenotype, namely CD8 enriched, minimally exhausted, and this is the product which now is optimized and can further proliferate in the patient. So as we think about OBX-115, this is our aspiration. We know we can make a drug product with a core needle biopsy. Our product is potentially compatible with lower doses of lymphodepletion than what is being used today. We are IL-2 sparing. You line up these three things together, and we ask ourselves, do I have an outpatient TIL cell therapy platform? We now have clinical data that we are seeing rapid onset of tumor control. We have the option to redose the ligand longitudinally to further drive deepening of responses. And we already have clinical data that we can induce CR. So we should be able to see transformational efficacy. You combine the outpatient nature and the efficacy, and our goal is to leave no patient behind who medically needs TIL cell therapy. So a few words on our clinical data. This is the schematic. The patient gets a baseline assessment, lymphodepletion, product, ligand administration, efficacy and safety monitoring. There is no IL-2 in the platform. A couple of words about the kinetics. On your left-hand side, you'll see that as the lymphodepletion is given, the absolute lymphocyte count goes down. The product is infused, the ligand is administered, and you see a massive expansion of the peripheral blood lymphocyte. The graph on the right shows that on most of these expansion points, the drug product is proliferating. And the drug product, as measured by the OBX-115 transgene, is persistent up to around day 42, at least in this patient. When we take these cells back to the lab and redose acetazolamide, these cells re-express IL-15. This is the data that we had press released in December of 23. 6 patients treated, 20 weeks of follow-up. The response rates are on the left. We got 3 out of 6 responders, 2 CRs, 100% DCR or disease control rate. In a few hours from now, update of this data will be presented by Dr. Amaria at AACR. This is a schematic of our multi-center study, very similar to the previous schematic that you saw. We are going to be requiring the ACZ redose at week 6 to be mandatory, and if the patient were to progress, they will again get a redose because we now have preclinical data that redosing of ACZ can make a meaningful difference. Just a few words on our next strategy. The cytodrive platform has the ability to control a protein of interest. So as we think about the next generation of TILs, OBX-115 has optimized the cell therapy characteristics. So we ask ourselves, can I combine it with yet another cytokine that has the ability to modulate the tumor microenvironment? The cytokine that we are focusing on is called LIGHT. It is tumor necrosis factor superfamily 14, or CD258. This poster was presented at AACR a couple of days ago. So stay tuned, because a combination of IL-15 and LIGHT may have advantages in tumors that are fibrotic in the stroma, the world of colorectals and ovarian cancer. So thank you for your attention, and I'm sure we'll get into the discussion later on. Thank you. Would like to introduce Regan Jarvis, CEO and scientific founder of Anoka. Regan. So Tom, why don't you come up, Charisma Therapeutics, CBO.
Make sure we have the right slides coming up next. Perfect. Well, thank you for the introduction, and good to be here with everyone this morning in Rome. And thank you to ARM for the opportunity to come and present, tell you more about Charisma and our engineered macrophage platform. Before we get into it, just draw your attention to our forward-looking statements. So when we think about solid tumor adoptive cell therapy, there are 3 key challenges. Essentially, trafficking to the tumor is the first one of those. T cells, NK cells are naturally excluded from the tumor microenvironment. The second challenge is if those cells do get into the TME, then they are rapidly shut down by the immunosuppressive tumor microenvironment, which is in part modulated, controlled by tumor-associated macrophages. And then lastly, solid tumors, unlike the majority of blood malignancies, are incredibly heterogeneous. We've already discussed this a couple of times this morning. So targeting a single antigen is probably not going to lead to a durable cure, and you need to address the multiple neoepitopes within the cancer. Our scientific founders at the University of Pennsylvania, obviously Penn has been a key center for the development of CAR-T. They'd previously developed Kymriah. They decided to take a slightly different approach, use a different cell. In this case, we work with the macrophages and now monocytes. The reason for this is that macrophages, through their natural ability to traffic to tumor, give us the opportunity to be able to use this as a Trojan horse type approach. We use their natural trafficking ability to deliver our payload with the macrophage into the tumor microenvironment. We engineer them to express a CAR. In the case of our lead program against HER2, we also engineer them to be pro-inflammatory, so the opposite of the tumor-associated macrophages, which are M2 or anti-inflammatory. Once within the tumor microenvironment, they will engage, phagocytose the cancer cells. They will also release cytokines and chemokines, warming up the tumor microenvironment. And then lastly, and really importantly, they are one of the only professional antigen-presenting cells. So they will chew through the tumor, and they will present out the multiple neoepitopes within that tumor for attack and destruction from the adaptive immune system. And it's really this unique triple mechanism of action which we believe makes them ideally suited to address the challenges with solid tumors. This is the typical side-by-side comparison slide versus T and NK cells. We do believe, obviously, in macrophages, that's our focus. It's not to say that T and NK cells don't have their role. Clearly, they've both been incredibly effective in blood malignancies, and ultimately, it's highly likely that we'll need to engineer all of these different cell types. They all have their own unique role to play within the immune system. But for the reasons I just mentioned, that unique triple mechanism of action, for the safety profile of our product, it doesn't require chemotherapy, unlike the T cell and NK cell approaches. In terms of the safety data we've seen so far in our initial phase 1 study, the product is very well tolerated. We've seen no dose-limiting toxicities, no severe CRS. The worst we've seen is some grade 1, grade 2 CRS, which has been transient and resolved with Tylenol. And then we've also discussed this morning the importance of speed in these patients. With our CAR macrophage product that has a 7-day manufacturing cycle, with our CAR monocyte that we've just taken into the clinic, we can manufacture that in a single day, so a very rapid manufacturing process. Our initial application of the technology is a CAR macrophage against HER2. We think HER2 is an ideal target for us initially. As a cancer target, it's not shared, it's not internalized. Obviously, there have been a number of new treatments coming to the market for HER2 more recently, but we still see significant unmet need there for a durable, lasting treatment effect. The initial application and the initial clinical study that we started a couple of years ago is for the CAR macrophage. This is an ex vivo autologous engineered macrophage. We engineer it using our adenoviral vector, Ad5F35. We recently started a combination of that CAR macrophage with pembrolizumab. In addition to that, we recently received IND clearance for the first ever CAR monocyte to go into the clinic. It's the same approach, the same CAR using the adenoviral vector, but there are a number of potential advantages which we'll discuss now. In the initial CAR macrophage data that we presented at the end of last year, we've seen very good feasibility. We were able to make large quantities, up to 3 billion CAR macrophage cells, very good purity, potency, good CAR expression. We saw very strong safety, as I said, no dose-limiting toxicities, no severe CRS. And essentially, we saw the mechanism play out. These cells are trafficking to tumor, warming up the TME, recruiting in T cells, NK cells, dendritic cells, other effector cells, and lastly, presenting antigen driving novel tumor-reactive T cell clones within the tumor microenvironment. The best overall response we saw in this initial study in this heavily pre-treated late-stage population was stable disease. So the next step for us is to see how we can go one better. We have two key strategies for that. The first is the combination with pembrolizumab. As I mentioned, macrophages will present to the T cells, they will recruit them into the tumor, but in the majority of these late-stage patients, we saw that they had an exhausted T cell phenotype. Now, the macrophage won't do anything to unexhaust the T cells, so we think the combination with pembrolizumab is going to be required in this specific patient population, and we started this combination study in the middle of last year. The other strategy we have is to move from a macrophage to a monocyte. The key advantage there is that we can significantly increase the dose. A macrophage is a non-expanding cell population, so the total number of cells that you can harvest with a single apheresis is the maximum that you can produce. Moving to the monocyte, we can significantly increase this, so up to potentially 12 billion cells from a single apheresis. This is some preclinical data on the combination of the CAR-M with pembrolizumab. On the left-hand side, we can see in this syngeneic non-PD-1 responsive mouse model that PD-1 alone does not have an impact. If we combine and the CAR macrophage as a monotherapy, we see approximately 30 to 40% of these mice are responders. If you combine both of the agents together, we double that response rate. If you look on the right-hand side, again, the anti-PD-1 on its own, we're not seeing much difference versus the control with blue cancer cells. If you put the CAR macrophage in, you see a significant increase, obviously, in myeloid cells and also T cells. But if you combine both of the agents, you really bring the whole immune system. A massive increase in T cells. We see NK cells, we see dendritic cells, and a lot more macrophage activation. So we really think this is going to be transformative. We're planning to share initial data from this combination in the clinic in the first half of this year. The second approach, as I mentioned, is to move to the monocyte. Key advantage here is that we can significantly increase the dose. So if we see safety with the macrophage, we see the mechanism, we've seen some activity, drug development 101, you ramp up the dose. So not only increased dose, we also see far better trafficking. Macrophages are big sticky cells. They tend to get stuck in the liver and in the lung. We move to the monocyte. These cells traffic far better, 40 times better in preclinical models. And then they're actually far longer lived, 10 times the persistence and actually slightly more potent cells. So when you put all of this together, you end up with around 2,000-fold increase in total exposure within the tumor microenvironment, which we are confident, obviously we don't know until we go into the clinic, but confident that this will significantly increase the activity within the TME. The final slide I'll share with you is a collaboration that we're particularly excited about. We signed this deal a couple of years ago with Moderna. Obviously, the first iteration of our technology is ex vivo autologous macrophages. We've spent a lot of time there getting that process as efficient as possible. But clearly, there would be a significant advantage to being able to engineer these cells within the body using off-the-shelf therapeutic. In this case, an mRNA LNP provided by Moderna, which is engineered to target myeloid cells in particular, deliver a CAR to them within the body, and essentially engineer the product in situ within the body. This would provide an off-the-shelf therapeutic, one-size-fits-all, that is truly personalized for the patient. And as again, we've discussed this morning, the benefit of that being able to democratize cell therapy, potentially take it into a far broader audience than we could treat with a ex vivo autologous approach. So I think we'll talk about this a bit more on the panel, but that was my final slide. Thank you. And with that, I'll introduce the final presenter, Regan Jarvis from Anoka.
Okay, thanks, Tom. It's nice to be here in sunny Rome, especially because there was snow on the ground in Sweden when I left home, so it's very much appreciated, the location. So I'm going to jump straight into it. Just talk about Anoka and our approach. We're a TCR-T company, and just looking at the transformative targeted cell therapies. When I say targeted, I mean modified to target specific antigens like CAR-Ts. And they have been incredible, transformative for patients where targets are available, and that's mainly hematological malignancies so far. Next generation of targeted T-cell therapies will be TCR-T, and it really opens up a new target space, and that's what I'll talk a bit about today. So there are two fundamental challenges. One is the fundamental barrier to entry, which is can you find the targets and can you generate the receptors, which is non-trivial. The targets are very complex, and I'll talk about that on the next slide, which you need to map and quantify and rank, essentially. There are immense numbers of these potential targets, and you need to make these fairly complex receptors. And the second barrier to entry is manufacturing, which everyone talks about in the cell therapy space, but a bunch of emerging technologies, of course, and just making a stable process at reasonable cost is always a challenge here. So we have built a platform on two dimensions. First is a deep tech approach to map the targets, generate the TCRs, optimize these TCRs, which we can do very cost effectively and very efficiently to access the very broad target space here. And the second is we've actually built our own manufacturing platform, which is the largest approved facility in the Nordics. One of the largest in Europe, we think, but we haven't looked into that so far. So just to double tap on CAR-Ts versus TCR-Ts to understand this a bit more. CAR-Ts, like I say, incredible targeting, retargeting T cell cytotoxic activity towards malignant cells with a chimeric antigen receptor. These are a cool workaround to T cell complexity. They're these Franken receptors, which is an antibody fusion with a bunch of TCR signaling domains. But you can only access about 10% of the proteome here because these Fabs are restricted to the cell surface of the target cells, the tumor cells. Not only are they restricted to the cell surface, they have to be quite high density, right? Because these CARs are fairly weak signaling compared to the genuine T cell signaling components as the T cell receptor. In contrast, the TCR looks at everything inside the cell. It opens up a window by looking at what are called HLA-restricted peptides, because all the proteins in the cell are turned over by the proteasome and ported to the ER, loaded into an HLA and presented at the cell surface. So TCRs open up 100% of the proteome, all this target space, which can be incredibly tumor selective as well. So we've deployed a technology to map that target space and make these TCRs. And just to look a bit more at the challenge here, the TCRs themselves, they're like antibodies, VTJ recombination. There's 10 to the 18 possible diversity in humans. There's a lot of them to sort through. And of course, they're cell receptors. They're not antibodies. They don't, you don't rank them on affinity. You rank them on activity. So you have to account for that. The second part is these HLA-restricted peptides. There's a lot of tumor-selective target sequence there. You heard Madan say that there's a lot of neoantigens that are not potentially shared. There are some that are quite shared, and you need to sort through those. And there's a diversity of the HLAs as well. Different HLAs and different people will load different peptides. HLAs are the most diverse genes in humans. So there's a lot of target complexity here. In talking about manufacturing, obviously, living cells as a product is just a basic challenge. You need a lot of specialist infrastructure. There's limited industry capacity, know-how, high COGS, and lots of new competing technologies coming through. And just to look at how we've set up the company in the last 10 years, we've built a biotechnology platform, which the differentiator there, it's all cell biology driven. There's no computational predictions, there's no biochemistry. Everything we do is a high precision cell-based assay. We've built a very large software ecosystem behind that, because that's powering us to do this in a very parallelized, very efficient manner. There is lots of target space to capture, and that's the game here, is how can we do it efficiently and effectively to get the most target and most patient coverage. We built a pipeline already of over 40 TCR-T assets, and we'll be progressing those into clinical trials end of this year. And of course, we established our own manufacturing plant near Stockholm in Sweden, and we've just announced recently a license of CRISPR-Cas gene editing technology to do autologous TCR-T in a gene-edited manner. So just to comment very briefly on the concept behind the platform, everything we do, as I've already said, is a cell-based assay, cell-based analytics. And that seems strange to say as differentiating. You were using cell biology to make cell therapies. But in the TCR-T space, it's really not the norm. People are using a lot of computational biology, a lot of affinity measures, biochemistry, phage display. Everything we do is cell display, cell selection. That's powered by a bunch of biotechnologies that basically allow us to compile these cell-based assays very precisely and very cheaply. And finally, that software ecosystem is basically like an enterprise software built by us for us to capture all the data, do the analytics pipelines, even does protocol development in the lab and implementation. This is I'll just flash up the pipeline. I'm not going to read through it all. Just to say our lead programs will be KRAS G12V targeting. That will hit clinic later this year. But there's a lot of target space. We're starting with 40. It sounds insane as a small biotech, but there is a unity of manufacture. It's a unity of mechanism of action. It's not a unique product at every line. And this is sort of owes to the fact that there's a lot of target space to sort through, and this is the first tranche of assets we're taking through. So just to look very briefly at how the platform actually works, we start with a target gene of interest, a tumor-selective gene sequence, like a KRAS mutation, a tumor cancer testis antigens, things like this. And we plug them into an antigen discovery platform. We basically program engineered antigen-presenting cells. We pull on the HLA, shoot them into a mass spec, and we actually quantify which peptides are presented from these target sequences on an HLA by HLA basis. We take those candidates into a multiplex TCR discovery from healthy donors, which works in just a couple of weeks to get our list of TCR candidates. And we pop those into a cell-based assay where we can back-validate to the map targets. TCRs are a gold standard, don't rely on the mass spec, and get some basic telemetry basically on how those TCRs are acting. And then finally, the heavy lift in the platform is about safety. TCRs are these very intricate cell receptors, and they have had clinical issues with off-target toxicities. And we have a three-step process. For those who know a bit about T-cell biology, it's like an ex vivo thymic selection, but they're all negative selections. There's no positive selection step in this. So we account for allo and cross-reactivity, the two major phenomena here in off-target TCR activity. And then finally, native TCRs aren't always going to cover the targets, these high-value targets that you need. So we've generated a TCR engineering platform, which essentially works as a single-step directed evolution of TCR potency. We can also clean up TCRs that are have a bit of target toxicity. But end-to-end, we can run this in a highly parallelized manner. We can go from interesting target sequence to a TCR-T asset in less than 12 months. This is skipping all that cool preclinical data. This is the bottom line data. First asset is the G12V targeting TCR and the HLA-A11 restriction. It clears tumors in mice really quickly. This is a G12V expressing lung model with luciferase expression. It clears this in just a few days with a survival advantage in the model. So the infrastructure around manufacturing, we've built this in a former AZ plant near Stockholm. It was certified last December by the Swedish regulators. It basically enables us to independently advance assets into clinical studies, and we've got control of all the technology coming through here. Like I said, we've licensed a gene editing technology, which allows us to do precision gene editing, single locus, single copy, which is quite nice. And more importantly, it's a technology that allows us to enables us to scale out. We don't need to make a viral vector every time. We just need to make a gene construct where we can reuse the editing reagents again and again. When you've got a pipeline of 40 plus assets, that's something that's forefront of our minds. And then we've got our in-house PD, QC, QA teams. Obviously, we're still assessing technologies to make this process better as we go. And just finally, big thanks to the team. We're about 115, 35 nations represented there in Sweden. Super fun place to work. We're based in Södertälje, which I still cannot pronounce correctly, I don't think. That's near Stockholm. So thanks for your attention. I look forward to the panel.
All right, let's get started. Great presentations. Madan, I'll start with you. So obviously Obsidian has been very focused on patient access. How do you reduce toxicity to improve that? As you think about your program design, what other additional patient access barriers have you defined and how are you addressing those?
Yeah, great question. I think if you think about from a TIL standpoint, the patient journey, the first thing is currently the standard of care is to get a surgical excision of the tumor because that's your raw material. And the question we ask ourselves is, can we decrease that burden on the patient and start the manufacturing with a core needle biopsy? And at least based on our early experience, it looks like our platform is compatible with the core needle biopsy. The second piece is that patients need a certain amount of lymphodepletion, which is still at the end of the day cytotoxic chemotherapy. And the question is, can we reduce that lymphodepletion further to decrease the symptom burden on the patient? And again, the early data shows that we're moving in that direction. Obviously, the biggest value driver for us is that we are IL-2 sparing, so that's where sort of most of the value proposition comes in. As we're thinking about the patient, it's very important to think about the impact on the treatment center. So as you make the product more safe, more differentiated, it's easier to administer the product for the treating physicians, the healthcare systems. So that's also an important part of the ecosystem. And at the end of the day, we want to make sure that all the services that we provide, the COI, the COC, the batch records, everything needs to be sort of digital so that it's easy on the entire ecosystem because all of that at the end of the day contributes to patient access. So that's the way we are thinking about it today. And obviously we want to, try and solve these problems in a more sort of a phase-specific criteria. So yes, we've got the eye on the end game, but we don't want to, necessarily solve everything too early on and sort of approach it in an incremental manner.
Thank you. So Tom, I'm going to go to you next. So you also talked about going outpatient for your program, and your approach is a little bit different, right? You're working with Moderna, you're using other technologies. Are there other potential benefits that we should be thinking about with your specific strategy? Does it help with duration of effect, response, anything you could share there?
Yeah, so this is a program, as I mentioned, we started a couple of years ago, leveraging our expertise in macrophage engineering and Moderna's LNP mRNA platform. One of the key challenges with mRNA, obviously, is it's transient, and if you were to try this with an expanding cell population like a T cell, you would rapidly lose that expression. We actually think macrophages are the ideal cell type to use for this in vivo LNP mRNA approach. And the reason for that is if you think about their mechanism, macrophages patrol the body, phagocytosing any foreign particles, any foreign matter. So you put an LNP in, the chances are a lot of it's going to end up in the myeloid compartment. In this instance, we actually use a myeloid tropic LNP that Moderna have developed. As I mentioned, that it's not an expanding cell population, so we actually have engineered the mRNA to get, typically you see a couple of days of mRNA expression. We now actually get 2 to 3 weeks of expression. And we think that is long enough for the cells to traffic to the tumor, to warm up the tumor, and then critically to present antigen to the T cells. And it's this presentation, this adaptive immune response that provides that lasting effect and lasting immune memory. So our hope is that this would drive a longer-term response against the cancer, not from the macrophages, but from the T cells over time. So hopefully getting around this issue of transient expression.
Excellent. So Reagan, coming back to TCR, so antigen escape, how should the field think about this?
Yeah, it's a question we always get. I mean, antigen escape is, well, therapeutic escape is a thing with every oncology therapeutic. You see antigen escape with CAR T, there's CD19 loss, various mechanisms at play there. TCR targets, or the HLA-restricted peptides, is a bit more of a delicate system. There's the whole HLA processing and presentation machinery that can be damaged. So in the first instance, we're taking the approach of selecting indications where it's very rare that you see loss of heterozygosity or damage to the proteasome or at the HLA loading machinery on the ER side of things. But ultimately, T-cell therapies are unique in the fact that they're directly harnessing T-cell cytotoxicity. So the aim really is to hit the tumor, hit it hard, and to what Tom speaks to, is recruit the immune system around that, essentially. If you can do that aggressively, whether it's a monotherapy in terms of the TCR-T, if it's a multiplex TCR-T like T-Scanner pursuing at the moment as a sort of an upfront program, or if it's in combination with other drugs or vaccines. I mean, this is the promise, is to really harness this cytotoxic effect of T cells robustly and quickly to clear the tumor before you can get these sort of snowballing therapeutic escape. Like I said, it happens in all oncology treatments, but we are aware that this HLA system is a bit more delicate than many others.
Thank you. So Tom, going back to Charisma's program, one of the things that you guys are doing is combining with Pembro. So very different approach. So could you talk through kind of how you think about your program design or study design or some of the challenges that you may have to face as you think through how you kind of execute on this?
Sure, absolutely. So this is a study that we initiated last year, combination with Pembro. As I mentioned, in this phase 1, heavily pretreated, late-stage cancer population, when they come into the study, they're pretty beat up. We've had patients on 10th, 11th line therapy that really have no immune system left. So we think for this particular phase 1 population, the combination with Pembro, where the macrophage can warm up the tumor, present antigen, recruit in the T cells, but then you need to give the T cells a boost. You need to put a checkpoint inhibitor in there. We think it's going to be critical. The two areas that we've certainly talked a lot about and challenges, generally, obviously, combination therapies have been a mainstay for cancer treatment for a while, but ultimately you're going to have to convince payers that your product is worth paying for, and in this instance that the combination of both of them is going to provide something in addition to what you'd see with Pembro alone. We don't actually think that's going to be such an issue in this particular patient population. HER2-positive cancer, the vast majority of these patients are PD-1 non-responsive. I think Pembro has a very narrow indication in gastric cancer in combination with chemo and Herceptin, but has not proven to be successful. So we do think, if we can move the needle for these patients with the combination, that regulators and ultimately payers, physicians would view that data as positive of the combination having an effect. I think the other key challenge generally with commercializing combinations in this space is, we talked earlier about just the cost. These are not cheap therapies, and you start to put 2, possibly 3, 4, who knows in the future together, cost is going to be critical. So we've done everything that we can on the cell therapy side to reduce the cost, and we've done that significantly even over the initial period of this phase 1 study. But that's something that we're going to continue to focus on as we move to scale.
Madan, do you see combination therapy in your program?
That's a great question. I would say we're sort of thinking about it in at least 2 buckets. For the melanoma and non-small cell indication, kind of those are the trials that we have ongoing. Patients who have already failed checkpoint inhibitors, I don't believe there is good preclinical data that combining it with cell therapy again will move the needle. So I'm a little bit sort of skeptical about that. The other advantage that we have is, as we study our product, the product has very minimal level of exhaustion markers. So trying to combine it with an anti-PD-1 does not sort of pass the sniff test. That being said, if you're thinking about your product in the checkpoint naive population, where anti-PD-1 is the standard of care, there it absolutely makes sense because that's the accepted standard. Also, you have a certain timeline from tumor harvest to giving the product back to the patient, so you could, quote unquote, bridge the gap. The next thing you have to think about is, how long do you continue the anti-PD-1 after you've given your product? Because if I were a patient and if I can finish my curative therapy or definitive therapy in as short a period of time, that adds value versus doing anti-PD-1, a cell therapy product, and continuing the standard of care anti-PD-1. So again, over there, we have to generate the data and see if you can abbreviate the post-cell therapy anti-PD-1. So those are the different sort of buckets. So I would say scientific bucket, regulatory bucket, and then a patient bucket.
Thank you. Same question for you, Regan. You already have 40 programs, so do you see combination therapy in your future?
There's a lot of opportunity there, right? But I think it's worth remembering that CAR-Ts and the clinical studies in flight for TCR-Ts are kind of combo therapies already. These patients are all lymphodepleted with Cy/Flu, basically. And it goes back to what both Tom and I mentioned, that sort of limits the possibility to recruit the immune system aggressively in those first days and weeks of the cell therapy administration. So this is something we're looking very hard at, is not drug treatment, but co-treatment with our other immune activating agents to help that CD4 compartment come around our CD8 product. At the end of the day, the targets I talk about, I skipped over it for brevity, but these are HLA class 1 restricted targets that are targeted by CD8 T cells, which are cytotoxic. There's a whole other universe of T cells, these CD4s that provide the help. You need both active to have a really robust anti-tumor effect, especially in the solid tumor space. And so we're looking very hard at how we can recruit that CD4 compartment and other immune cells around our product. But that's probably not an anti-PD-1 for many of the reasons that Madan had referred to. But yeah, we have a lot of opportunities, a lot of targets, a lot of products that we have to put through it, but we're taking it step by step, right? You need to start with a monotherapy. If you're going to do this, you have to open a separate arm to do the co-treatment. But first and foremost, I think we should, everyone working with cell therapies, think about how these are already combo therapies with the Cy/Flu and how that's damaging the opportunity to recruit the immune system avidly around that, around whatever your therapy is, TILs, macrophage, or T cells.
All right. So this next question is for all of you, and I'll start with you since you're closest to me, Reagan. So if you think about the recent event, if you will, by the FDA around the safety around cell therapies, and knowing that all your programs involve modification of the cells, what are you doing to ensure that integration events don't become a compromise in terms of patient safety?
Well, I mean, let's see how that data plays out, but it's likely to be random insertional mutagenesis with the gamma retrolentiviral vectors used to manufacture those CAR T products. First of all, cost benefit. The benefit far outweighs the risk there of these relatively rare transformation events that have been observed, and the FDA has slapped this label on the CAR T products. Now, getting away from viral vectors isn't part of our game because of the number of products we want to manufacture and deploy to clinic in a timely and cost-effective manner. So gene editing is the approach we've taken. That gets away largely from the random insertional mutagenesis, so that's a plus. Other companies, both in the CAR and the TCR space, are taking transposon approach, which does not get away from that, but does have the cost and timeliness benefit. So it really depends on the platform you're using at the end of the day. We've chosen a platform after doing a lot of diligence that is, it's a very stringent cutter. It has a long PAM sequence for those who know about CRISPR-Cas systems. So we're very confident on very clean site-directed integration. I would flag, if you're gene editing, right, you have to be careful about the recombination events. It's a different type of genotoxicity of worms you're opening. But like I say, you need to be careful on which cutters you select, which guides you select if you're using a Cas system, how you assess that recombinational genotox, which has a potential in all these products. But it's really about front-loading those assessments and making sure that the benefit outweighs the cost, or the risk, I should say.
Madame?
That's a great question. I echo all the sentiments that Reagan mentioned over here. I think a few things that I would add is that when you're transfecting peripheral blood T cells versus mature T cells, which are typically in the TIL environment, I think that's another layer of sort of inherent safety, that their transformation potential is low. That being said, we are very cautious. We do extensive insertional mutagenesis assays, prove to the agency that we are meeting that high bar. And at the end of the day, this is a risk-benefit ratio. The patients that we are attempting to treat, they obviously do not have great outcomes today. And I think as we've seen in the world of CAR T cell, the US FDA has clearly mentioned that, at least based on the current data, the benefit is still outweighing the risk, and patients are continuing to be treated. So obviously, we'll continue to watch it very closely. These patients are going to require long-term follow-up, and we'll see how the data shakes out.
Anything to add?
Yeah, so very straightforward for us. We use an adenovirus, so it doesn't integrate, and the approach with Moderna, obviously mRNA, so transient expression. But agree with the comments. I think in cancer that this probably won't be an issue, perhaps as you look at cell therapy in some bigger, broader diseases, lupus, autoimmune disease. Perhaps they could be more concerned depending on the patient population, but I doubt this will have a significant impact on cancer treatment.
Thank you. So I'm going to pivot a little bit. So all of you have multiple programs or opportunities in your portfolio. I think what the audience might be interested in knowing is, how do you think about prioritization? So again, I'll start with you, maybe Reagan.
Well, first and foremost, it has to be a good target. I mean, this is a very large consideration when we're going after KRAS. It's a foundational driver mutation. So you can knock the legs out of that, you knock the legs out of the tumor. I guess the second key consideration for us is really about how do we integrate our cell therapy into the current standard of care. You're not going to be able to do first line, obviously. It's experimental therapy. So what's the best way that we can select a target that matches an indication where we have the earliest line treatment possible, the patient's in good condition. It's an autologous product we're manufacturing. Those T cells need to be in relatively good condition as well. They don't want to be beat up like various chemos or other agents. So that's really when we prioritize it. It's that combination of is it a good target, and is there an indication where the current standard of care makes sense for us to slot in at an earliest possible phase for a phase 1/2 trial. I would say it's as simple as that. It's not simple to find a good target in combination, but those are the key considerations for us.
Yeah, it's a constant sort of reassessment of your programs. I think you've got to start with what is the addressable market, what's the competitive landscape, what are the regulatory hurdles, and how is your product sort of, where are you on that journey? Once you take a decision, stay focused. I've been a treating oncologist in my prior life, and sort of I have a common motto: uncontrolled growth in the body is cancer, uncontrolled growth in your pipeline is cancerous, right? So you have to stay focused. You need to know what resources you have in your organization and stay focused. So currently the way we are approaching it is we are staying sort of on the left side of the Schreiber's plot with the high TMB tumors with melanoma and non-small cell lung cancer. As we develop the next generation of assets that are scientifically designed to go into colder tumors, that's where we'll move towards sort of the middle and the right side of the Schreiber's plot. But until that happens, stay focused. It's melanoma and non-small cell.
Excellent.
So yeah, we certainly look at exactly the same factors that the other two panelists have mentioned: commercial viability, unmet need, scientific rationale. The other question we always ask ourselves, so we've been an oncology company through pretty much all of our existence. We recently moved outside of that and initiated a program in liver fibrosis. And the question we ask ourselves every time is, why cell therapy and why macrophages? If you can do this with a small molecule or a biologic, why would you go through the complexity, the expense, the time it takes to manufacture an autologous cell therapy? And the reason we believe in engineered macrophages for solid tumors, liver fibrosis, is really that they are multifunctional. So you can do multiple different things with one cell therapy product. In solid tumors, the phagocytosis, modulating the TME, presenting antigen, driving this immune memory. In liver fibrosis, resolving the fibrosis, dampening down the inflammation within the liver, and then macrophages can also drive regeneration of the hepatocytes within the liver. So if you can achieve multiple different things within one product, I think you have a value proposition versus those other modalities, and that's how we tend to think about this.
Thank you. So Tom, I'm going to stay with you for a little bit. So we haven't talked about manufacturing. So some of you talked about going outpatient, bigger populations. So as you think about the manufacturing scalability, feasibility, what challenges do you kind of foresee down the road in that regard? So Tom.
Sure. So I've focused solely on cell and gene therapy for about 11 years now at the University of Pennsylvania, a number of companies, and it's always the biggest challenge, whether it's cell therapy, gene editing is manufacturing. At Charisma, we've really had a laser focus from the beginning, building a really strong, experienced team. A lot of the folks who had worked on Kymriah or other cell therapy products that have been commercialized, coming in, starting to build the processes, understand what do we need to do to scale this. And a big focus for us over the last few years has really been on trying to get down the cost of goods, even at this early stage. So I'll give you an example. For the adenovirus, when we started the phase 1, cost per patient was $60,000 just for the virus. Clearly not sustainable. The team have now got that down to $5,000. As we move from the macrophage to the monocyte, we're going to a one-day manufacturing processing time with the potential to make this fully automated closed system. So you could have a distributed, regionalized delivery system here, very rapid, totally automated. Ultimately, where we're trying to get to is with an autologous ex vivo engineered product with a cost of goods which is not too dissimilar to where some of the T and NK allo companies are talking, sort of $35,000, $40,000. And we think that that's critical as you move into some of these bigger populations in solid tumors. So that's been a big focus for us.
Madan?
Yeah, great question. I think it's plan, plan, and plan, right? So you have to, again, be your sort of state specific. You know that your process will iterate over time. You need to have an eye on that and take those steps of process iteration through the lens of your clinical development plan, your regulatory plan, the resources that you have available, but you need to really know what that end game looks like. I think it would be a tragedy to reach to the end game and then have a process that is not scalable, because the delay at the back end is hurting the patients, hurting the bottom line, and gives you several years of delay. So the way we look at it is we know what that end game is going to look like. We know where we have to keep on sort of closing the processes, making it sort of scalable, and you start putting kind of getting the wheels sort of turning earlier on. And at the end of the day, this comes down to talent and expertise. You know, hire the best talent that your budget can tolerate, and get the smartest brains to work for you. I think that's what it really amounts to.
Great. So Regan, I see you nodding your head. Do you agree?
Yeah, wholeheartedly with everything said. Yeah, I would say iteration is the key word there. You know a cell therapy process is going to iterate through the life cycle. And there are two aspects from our point of view. The first is the biology. Have you got the best reagents? Have you got the best approach? Are you delivering those reagents to the cell in the best possible way? You know, in a gene editing approach, there are more components to deliver to the cell, you know, mechanically or otherwise, and that's something we have a big focus on. And secondly is the engineering component. Can you automate? Can you close systems? You know, how closed are they really? Do you have to uncap a tube for a piece of reagent? And you know, those things are all in our consideration, and that's why we're doing the manufacturing and the process development in-house. So we've got those close to our chest. We can iterate these in response to, you know, stage-specific requirements, of course. There's a lower bar here in Europe for phase 1, 2A on even reagent quality, for example. And then, you know, from our manufacturing facility standpoint, every process is in a separate suite. So we can change control those independently. It's not yet, of course, a single box solution like a Solaris or a long cocoon or something like this, because we think we need the flexibility of those change controlling independent pieces of the process in the meantime. And that's easier to do if it's an independent process step and an independent suite in the clean rooms. But yeah, it's, again, you need talented people to do this. We solve that with a 3-step process. We have a hardcore R&D team, tech development that does the assessments, transfers it to the process development team, and then it finally reaches the GMP team. So that's how we assess, implement, pass or fail that, then into the GMP. And we'll be doing this continuously as part of the R&D effort.
So a lot of what you do is very expensive. So I want to talk about funding a little bit. So congrats, Madan, on your successful Series C just last week, I believe. Anything you could share maybe to the audience, you know, who are kind of in the same shoes, you know, learnings, insights, how do you kind of get to that next round?
Yeah, it's great, and thank you for that shout out. We're very pleased with the outcome. I think the way to look at it is have a fantastic pitch deck. Don't scringe on that. 2 is make sure that the story you're telling is accurate, that you have the conviction in your story. Once you've written your story, then start thinking like the investor. Let's at least give them the benefit of doubt that they're there to help you. So allow them to help you. Listen to them. Don't push back. It's a dialogue. I think, at least in my interactions, I've found that I learn a lot from the investors. It helps you sort of refine your pitch. And again, you know, they are also looking for the end game. You are looking for the end game. And the end game is helping patients. So if you can start aligning on that earlier on, and you can help articulate that vision, often when you're meeting with the investors the first time around, you get a brief 30 minutes. And as we've seen in these presentations, if an investor is not a specialist in cell and gene therapy, it takes a while to explain kind of your platform, your nuances. So sort of refine that as much as possible and show them sort of that vision as to what your next inflection points are going to be. So again, it's a journey. It's not an event as such. And, at least for me personally, the board at Obsidian has been fantastically supportive, leverage the connections, and, talk more, talk frequently, be out there.
Great. Anything to add, Reagan, from your perspective?
I mean, hiring talent, maybe Madan wants to do our Series C later this year. I mean, it sounds like you know what you're doing, but not really. I mean, I just agree that you need to tell a crisp story. I mean, we've raised about $150 million in financing, and we'll need to do that again to get therapies into clinic. I mean, this is an expensive sport, as our chairman says. So it's good story with conviction, and be ready for left field questions.
Yeah, I'd echo those comments and the previous comment I made in terms of why your particular technology for this application, and if it's a cell therapy, which is always going to be expensive to take forward and get into the clinic, I think you have to have a really compelling reason why your technology is the one to do this. And then just understanding where the market's at. Clearly, 3-4 years ago, you could go out with a pitch deck, a bit of mouse data, and you could raise a ton of money. I think now investors are in a very different, probably more logical place, and particularly in oncology, where the preclinical models just aren't that predictive. Recognize that being able to drive towards clinical data and clinical validation is really critical, and being able to do that in a cost-constrained environment, which we're in now. So that's what we certainly think about.
Thank you. Well, we're at time, and we certainly don't want to stand between you and your lunch, so please join me in thanking our fantastic panelists, and thank you for being so generous.